It's my pleasure to be here today. I'd like to begin by introducing my co-moderator, Dr. Ehsan Nobak, Assistant Professor of Medicine with George Washington University, who will be conducting the Q&A portion of the session of today's panel. Today's speakers include Dr. Robert Starr, Director of the Division of Kidney, Urologic, and Hematologic Disease at the National Institute of Diabetes and Digestive and Kidney Diseases, NIDDK, and I'd like to see that on your business card, at the National Institutes of Health. Dr. Starr is an exceptional civil servant and a recipient of the AAKP National Public Service Award. Next, Dr. ELISA Thompson, Clinical Team Leader with the Division of Cardiovascular and Renal Products at the Center for Drug Evaluation and Research with the U.S. Food and Drug Administration. Dr. Thompson is a highly respected expert recognized for including patient insights in clinical trials. And completing our panel is Ms. Peggy Tighe, Principal of Government Relations and Legislative Affairs with Powers Law here in Washington, D.C. Ms. Tighe represents the policy initiatives of the American Society of Transplant Surgeons and is also a recipient of the AAKP Congressional Advocacy Award for her work to increase patient access to organ transplants and for vigorously defending the principle that patients should have full choice for their health care decisions. Dr. Starr, I welcome you to begin the panel.
Thank you very much. And I'd like to thank the George Washington University and the AAKP for inviting me to talk a little bit about what we're doing in patient-centered kidney care. And I'm going to be talking to you about a very innovative, difficult, and frankly audacious project that we think is actually essential to move the needle forward. And I have a few disclosures. I don't want to belabor this point because I suspect this was discussed a lot yesterday, but in nephrology, we think we have two disorders, acute kidney injury and chronic kidney disease. And the white space here shows the treatments that we have for them. For chronic kidney disease, we have and have had for 30 or 40 years ACE and ARBs, and probably will be soon appearing SGLT2 inhibitors. In the case of acute kidney injury, we frankly don't have very much at all in terms of treatment. We have fluids and tincture of time. So the white space, I mean, of course we have dialysis, which is a rescue therapy, but we'd really like not to get to that. The white space, if your drug company shows that there's lots of opportunity in this area to move forward, if you're a patient, it's frankly depressing. And for the patients in the room and on the internet, the punchline for all of my slides will be shown at the bottom right. This was suggested by one of the participants who's part of the KPMP project. Now, what patients want to know, and I think we've heard bits and pieces of this throughout the meeting, is what do I have? What's going to happen to me? What can I do about it? And what can it mean for my family? They prefer not to be put in a big bucket and get the treatment that everybody else gets, but be given some kind of precision medicine approach to get the right intervention for the right patient at the right time. Now, I forgot this slide at home, but if you can imagine the Star Trek tricorder, where you scan the patient and figure out, okay, what do you have, and immediately delivers the therapy. That's one vision of the future. We're not quite there yet, but with this Kidney Precision Medicine project, we think will get us the beginnings of it. So why do we need a Kidney Precision Medicine project? These two diseases, acute and chronic kidney disease, are currently defined by serum creatinine and urine protein, and we've had many fights about how to do that. But what's more critical is that they fail to cluster patients into distinct mechanisms of disease. So if you're heading for a precision medicine approach, you need to know what you actually have in some smaller category, not just what the larger category is, in order to be treated properly. The other problems are the animal models don't replicate human disease phenotypes, plural. In fact, we're talking about phenotypes too. We're not talking about acute kidney injury as a monolithic thing. We're talking about different subtypes of acute chronic kidney injury. Similarly, we're talking about subtypes of chronic kidney disease, so it's really plural, chronic kidney diseases. The animal models don't replicate these phenotypes. They don't allow the drug companies to prioritize hits. We don't understand the molecular events that occur in our patients, and we can't prioritize the targets. And we think that this is largely traceable to a decision that nephrologists made about 40 years ago to stop biopsying patients with kidney disease because the biopsy really didn't provide any additional data to help the patients. Could you imagine treating a patient with cancer without a biopsy of the tumor? I don't think so, but that's what we do in kidney disease. What we need is human tissue in order to improve human health. So the goals of the Kidney Precision Medicine Project are to understand and treat human kidney diseases, not animal diseases, human kidney diseases, plural. This will be done, as you'll see, by ethically and safely obtaining kidney biopsies. These are research biopsies from participants in the study who have common forms of acute and chronic kidney diseases. We will use this to create a kidney tissue atlas that I will describe, and that atlas will then be useful for identifying which cells are causing the problems, what pathways within the cells are causing the problems, what new targets are there, and this will help the community and drug developers to create therapeutic interventions for our patients. And this will stratify patients into subgroups and develop these individual treatments that we're talking about, improving the knowledge base of nephrology and improving two things. One, desperately needed for our patients is the pipeline of drugs, but we think this enhanced scientific knowledge will also bring more people into nephrology, which we also need. So the idea here is to use kidney biopsies to change patient management and improve the health of patients with kidney diseases. This is a large effort. At the bottom are the 100 to 120 people that are involved in this project, including about 20 subject patient participants, at least two of whom are here, Richard Knight and Paul Conway. I want to thank you, and the others, as I will talk about how the patients have helped push this project forward. The slide also shows the scientific organization. There are 6 recruitment sites that will be recruiting patients, 3 with acute kidney injury, 3 with chronic kidney disease. The tissue obtained will be interrogated by the tissue interrogation sites, and all of the clinical and tissue data will flow into the central hub. And I'll be describing these components as I go forward. Now, the patients have been involved from this project from the very beginning. The first scientific conference that we had before this initiative even was shaped, we had a group of patients that helped direct us what to do. They also go to all of the meetings. I sit next to them and help translate the scientific jargon into something the patients can understand, and they push the physicians to do the right things, to keep the goal of this in mind, which is understand human kidney disease and come up with better treatments, not just publish a bunch of papers or create an atlas of all the cells in the kidney. One of the things that we learned very early on, and no surprise, is that the participants were concerned about the research kidney biopsy, and actually 3 parts in particular. First of all, is it ethical to do that? We discussed this with the patients as well as a variety of ethicists, and came to the conclusion that as long as we were using the information to advance general scientific knowledge, and we're doing it thoughtfully and carefully with all the quality control metrics that I'll talk about, that it is ethically permissible to do a research kidney biopsy, as long as there's informed consent. The patients were also concerned about safety because there is a risk in the kidney biopsy of bleeding and a very small but palpable risk of death, and this needs to be completely disclosed to the participants in the study. And they were also concerned about the cost. Well, what is it if they have a complication from the kidney biopsy? Who's going to pay for it? And one of the things that they forced the study to do, and the study did it, was signed about a month ago, was to get so-called no-fault harm insurance that would pay for frankly, routine complications of the kidney biopsy. We pay for the medical costs. We're still working on paying for compensation for lost work. We're not quite there yet, but we did figure out this first piece. This is the first study at NIH to have done this. So kudos for the patients for forcing us to do the right thing. What the patients want is that the kidney biopsy will provide enough information so that it will become not just a research biopsy, but it will become a clinically useful clinical biopsy that will guide what do I have, what's going to happen to me, what can I do about it. And this is the culture change that I'll talk about. So for the scientists in the room, we will be assembling cohorts of patients with acute and chronic kidney disease. Shown here are the sort of approximate inclusion-exclusion criteria. The punchline here is that these are patients with the common forms of acute and chronic kidney disease, not the odd ducks that are getting biopsied right now. And that we've insisted on very strict quality control. Everything will be done exactly the same way from site to site. The sites have practiced doing kidney biopsies on oranges, apples, pears, whatever, to make sure that they handle the tissue properly, because it's actually quite complicated to process this tissue properly. And patients will be followed very carefully. These common protocols will increase the rigor and reproducibility of the study, and this is what we need as we think about doing research kidney biopsies on participants and collaborators. Now, one could guess that the technology parts of the study are going to be difficult, and I will talk about that in the next few slides. But before that, I want to show what I think is actually a harder part of the study, and that's the culture change that we need. Oops, sorry. Something didn't come through. We need the biopsies to impart clinical information, and that clinical information will be then used by the pathologist to sort of rebucket patients into the new correct buckets, not the ones that we've been using in the past. We hope that this will potentially blow up the way that we think about kidney disease, acute and chronic kidney disease, and that eventually what will happen over time, and I apologize, this slide isn't projecting properly, is that the patients who are told you have kidney disease, they're going to go to the internet, they're going to see, oh, the internet is saying I should get a kidney biopsy, and here's why. They're going to go to the nephrologist and push the nephrologist to do the kidney biopsy, push the pathologist to incorporate this new information from KPMP to properly interpret that biopsy, thus yielding them the precision medicine approach that we think is needed here. These are the culture changes that will be, frankly, more difficult than the technology. So let's talk about the technology. This is what a kidney biopsy looks like right now. In the future, we hope it looks something like this. Now, in full disclosure, 3 of these 4 pictures were randomly picked off the internet, so they're worth what they're worth. One of them is real. It's the one on the upper right, which is from Andy McMahon's lab. But what we want to happen is we want to harness all the new technology that I'll be talking about, where we're able to analyze single cells, peer within the cell, ask what is that cell thinking? Is it, first of all, what kind of cell is it? We won't be doing the usual thing of pointing, well, the proximal tubules are over here, the glomerulus is here, the thick ascending limbs are somewhere over here. We'll have the markers to show where we are in the kidney, what are we looking at, where are the cells, and the areas between the cells, the interstitial cells. We'll be able to paint the cells, all the structures in the kidney, and then say this cell is sick, this cell is healthy, this cell is undergoing productive repair, this cell is undergoing unproductive repair, this is a cell that's causing fibrosis. Then find the activated pathways that are activated within the cells that are causing the damage to the kidney, and start looking at pathways and begin to ferret out projects. So the whole goal here is to understand the heterogeneity between the regions of the kidney, between the neighboring cells. And some of this will require not just 2D imaging, but 3D imaging, because in the glomerulus and interstitium, the cells are packed so closely together, it's hard to see on a single cut what's going on. You need to look at a stack of images. What this is about for the patients, this is how to find useful information, clinically useful information from the biopsy tissue. So what we will be doing is producing a kidney tissue atlas. What's an atlas? It's a collection of maps that tell you what's going on in the world. It can show you a variety of data, geopolitical, social, economic status. Similarly, the kidney tissue atlas will have multiple maps showing the histology, showing what cells are where, showing the pathways that are activated, and all the things that I've talked about. And that the content of the atlas will greatly improve over time as our technology changes and improves. The best example I can use for how we're going to do this is, let's say you're going on a hike and you come across this leaf and you wonder, well, what is it? So you look it up in a leaf atlas, and leaf atlas tells you, well, this is the stem, and here's the leaf, and here's where the chlorophyll is. That's kind of interesting, but not what you really want to know. What you really want to know is, what kind of leaf is this? What will happen to me if I touch it? And what should I do about it if I touch it? And there you go to your leaf identification card that tells you this is poison ivy. If you touch it, you will itch, and you better start taking steroids if you touch it. This is the kind of information that will help make decisions. The first part is easy, the visual display of spatial information. This second part is actually more difficult. I'm going to skip over these parts. So there are two major deliverables to this project. One is the tissue atlas that will take what we currently see now in a routine histology. We'll be doing digital pathology. That's better. But you really want to know is what parts of this tissue are inflamed, what parts are fibrosed. And here the group has developed a bunch of machine learning algorithms that can do this on the fly in real time. It can show what parts of the kidney are diseased, what parts are not. And what we want to do is not just figure out where things, using the known patterns that we already know about, we want to figure out what are new patterns of injury that we can't see with the human eye, but the machine learning algorithms will help us identify. All of this has to be tied to the patient data that we're collecting. We'll be collecting longitudinal data over time to see what happens to the patients, and then link the kidney architecture to the clinical outcome data, be able to predict meaningful outcomes from the kidney biopsy. Right now, biopsies are used for a diagnosis. We want to push that to prognosis, what will happen to me, and push that to therapy like is possible now in the cancer space. Kidney's not there yet. And this will evolve over time. The second part of this project, and this is what distinguishes it from all other Atlas projects out there, is that there is a deliverable that's a clinical assay. We're not just doing to understand what's in the kidney and how the cells are working and what they're thinking. We want some assays to help our patients understand what they have, what's going to happen to them, what they can do about it. These assays may take the form of a fruit smoothie, a fruit tart, or a fruit salad. So let me explain briefly. You can take the kidney and grind it up in a blender like you would with a fruit smoothie, and that will tell you the percent composition of what was in there, but it won't tell you where things were. The fruit salad, on the other hand, will tell you what cells, what pieces were in the kidney, and you pick and cut them, but it won't really tell you where each piece was. It will tell you what was in the piece. The fruit tart analogy is that you bust the tissue apart, get some markers, and then use those markers to answer what was where. The kidney is anatomically complex. We think that's the kind of information that we need. For the researchers in the room, this is using single cell, various techniques to bust the tissue apart into single cells, examine each cell individually, and then cluster the cells into groups shown here. This is actually quite difficult and quite costly, but we will be doing this on these research kidney biopsies to get the markers that then enable the other parts here. The middle part is to use these markers on the sections to ask what is where, and that's the assay that I think will be most important. This is the kind of assay that's being used right now in breast cancer with her to understand what patients need what kind of therapy. The other possibilities are we'll be able to grind up the tissue and use bulk RNA sequencing with some deconvolution methods. what that means is that you can do this quickly and more easily, and that's what's being used in cancer patients to decide whether to go on checkpoint inhibitors. So all three of these different assays are feasible, and we're going to be looking at all of them to see which ones work best in which patients. This is a very fast-moving field, and disruption is likely as we move forward. So one of the things we've incorporated in this project is versioning, of making sure that we keep track of the blocks of tissue so we can go back as the assays improve over time and revisit patients that were studied early on with the later assays. So the challenging parts of KPMP and unique parts. Well, the unique part is that when patients have been involved from the beginning, they've pushed us to make sure that this study is done under the highest ethical situation, that it's safe, and that we have rigorous quality control standards in place. These are research biopsies, not clinical biopsies in patients with common kidney disease, not rare diseases. We need to follow these patients for long periods of time. This is not a short 2-year project. This is a 5 to 10 to, frankly, 15-year project. Then we need these large cohorts in order to get the personalized medicine metrics needed. We're going to be spending a lot of time finding out what we don't know. This is the so-called dark matter, because that may be where the answers are. The parts of the kidney that we haven't bothered looking at for 40 years may be exactly the ones that tell us, here's what's going on. We need to create routine clinical assays that will be used in routine clinical practice, and it's this project that will be making these assays, not something else that happens afterwards. And then we need to develop new patient characterizations or endophenotypes to find these patients over here look like they have CKD type A, type B, type C, et cetera. And these are in each of these individual clusters, the therapies will be different. We'll begin our version. The biopsies will start in about a week or two. We finally cleared all of the safety hurdles, ethical hurdles, and quality control hurdles. So we're really excited, and it's finally time to get started. Our journey begins. This is version 0.5. We need to get to version 5 probably. And what I'm going to do is thank AAKP, thank all the patients here, all the patients on the internet, because we need you to be part of this project, to find out where your local center is that's involved in this. And please, please help us move this project forward. Thank you very much. And I guess I'll take questions at the end.
As you heard from Ed, my name's ELISA Thompson, and I am for a long time, I was the clinical team leader for the group in our division that regulates the renal products. Right now, I wear 2 hats. I'm still the team leader for that group, but also the deputy director of the division of cardiovascular and renal products. And I, like Carolyn, I thought I'd show you where we actually sit, since I know this is sort of a broad audience and an international audience. The FDA, the Food and Drug Administration, is nearby. It's in Silver Spring, Maryland. It is quite a large campus, and as Carolyn noted, there are a number of people who work for the agency. Where I sit is the Center for Drug Evaluation and Research, and our group is tasked with making sure that safe and effective drugs are available to improve the health of the people in the United States. The group that I'm in the Office of New Drugs, provides regulatory oversight for investigational studies during drug development, and we also make the decision about whether or not drugs can enter the market and be available to patients to use and prescribed by clinicians. The session that I'm part of is called Government Agent of Change, and I had to Google that. It wasn't necessarily a term I was so familiar with, and when I Googled Government Agent of Change, I didn't get so many hits, and then I just tried Agent of Change, and I got more hits. I think Carolyn in her talk about CDRH did an excellent job of describing how CDRH has been an agent of change. I'm really going to tell a little bit of a different story, which is more a story, I think, of how the community has come together to really change the landscape, at least of drug development for a number of kidney diseases. And then I want to focus on really just one initiative at the FDA and at the Center for Drug Evaluation and Research at the FDA, and that is really patient-focused drug development. So again, where I sit is on the drug side, not the device side, and have for a number of years worked within the development and regulation of drugs intended to treat kidney disease. And I would say that since coming to the agency over 10 years ago, the landscape has really dramatically changed for drug development for kidney disease. In contrast to 10 years ago, I think there is now, I think, a great sense of hope. And this hope is born from some recent successes and also a greater interest on the part of industry in developing treatments for kidney diseases, or at least some types of kidney diseases. And I think there is a sense, I feel it at least, but I think there's others in the community who feel it as well, that we're really entering a new era for drug development for kidney disease, or kidney diseases, a very exciting area, and that really the dog days are over. This period of stagnation, inactivity is over. And so just to point out to some of you, many of you have probably seen this when I refer to recent successes. What am I talking about? One of these I'm talking about was described about a month ago in this publication that came out of the New England Journal of Medicine, showing that canagliflozin, what's referred to as an SGLT2 inhibitor, actually slowed progression of kidney disease, reduced the risk of progression to kidney failure, and also improved cardiovascular outcomes in patients with diabetic kidney disease. The associated editorial for this in the New England Journal, often editorials offer some criticism. This one was very hopeful. Overall, the importance of credence, which was the name of the trial, a well-done and large clinical trial, cannot be overstated. So again, the idea that really we have really hopefully have a breakthrough here in terms of a therapy for essentially a disease that we hadn't had a discovery for, or a therapy for over 15, 20 years ago. That was when the ACE inhibitors and ARBs were approved. So, as I noted, I think there is increasing interest in this space on the part of industry. And so why do I say that? Over the years, I've tried to keep track of drug development in our renal disease programs, both the number of requests that come in for meetings to discuss potential drug development programs for kidney diseases, as well as our applications and where they are in their phases of development. And I would say that on a very positive note, industry is keeping us busy, and it's keeping us increasingly busy. I would say that there are a steady flow of meeting requests coming into the division to treat kidney diseases. We're also seeing it move beyond just a request for a meeting, but sponsors then actually coming in and submitting their applications to start first-in-human trials, or to take a drug that's been developed for another indication and start testing it as a treatment for a kidney disease. I would also say that if you look at our drug development programs in the kidney space, many of them are progressing to later stages of drug development, getting past those early phase trials that are showing you proof of concept and making it to those phase 3 trials, which are the pivotal efficacy and safety trials, which ultimately support a marketing application and drug approval. I think another metric of industry interest in this space is the increasing number of meetings being organized by the community related to drug development for kidney diseases and initiatives in the space. And we just thank, again, the meeting organizers and for also including us in this discussion of global innovation, because I think finding new drugs to treat kidney diseases is certainly part of that innovation. Beyond these metrics that I see, I think there's also a shift in what the community realizes it needs to do. And so this is from the website put out by the NephCure Foundation, a foundation that has done very important work to try and advance the development of treatments for a number of kidney diseases that are associated with the nephrotic syndrome. These diseases typically affect children as well as adults. And in that group, which for a number of years was focused on really getting some of the science around the diseases understood, has now shifted to really try to develop an infrastructure to allow the conduct of the clinical trials that all these industry sponsors are interested in doing, because they realize that unless they can actually get the patients into all these studies, industry is going to walk away. And so here you see what they've developed, and it's still being developed even further. You can go online and find this Kidney Health Gateway. It's a gateway for patients to find out about clinical trials that they might be able to participate in. It's also serving as a registry to sort of understand these patients better as well. And as noted on their site, nephrotic syndrome patients have a great reason to feel hope, because today there are more clinical trials for nephrotic syndrome conditions than ever before. And so again, today the real challenge is actually just filling those trials. So how did we get here? It really takes a village, and I think at the center of that village, though actually it's, I guess, on the left upper hand corner of my slide is the NIH. It's Dr. Starr's group in the basic science research that they funded, as well as a number of the observational studies that they funded to enable us to have a better understanding of a lot of these diseases, how they progress, who is at greatest risk of progression, as well as biomarkers, things that we can measure in the blood, in urine, imaging that we can do to better inform our understanding of who is more likely or less likely to progress, as well as really tools that we can use in drug development to get early readouts of efficacy, et cetera. There's a number of other people who have played an important role in this space, and I can't put them all up here. Certainly those who have advocated for funding for these important NIH initiatives. A number of other patient advocacy groups have also done a lot to really move the needle in terms of addressing some of the obstacles to drug development in the kidney space. And some of these obstacles center around what endpoints we should be using to establish the efficacy of our therapies. Obviously, the outcome we all want to prevent is progression to kidney failure and the need for dialysis and the need for a transplant. But, the time course for progression in many of these diseases is very long, which means you need to find or identify earlier indicators that are reliable indicators of a treatment's effect on, progression to kidney failure, so that you can really accelerate drug development. And so here I just highlight a number of these. Also want to note that we got where we are because of the important work that these groups are doing, and I think that we will get much further based on additional efforts that are underway via these groups to really establish, and I don't know if that's me, an infrastructure, so I apologize, this infrastructure to really get these trials done, and really their efforts to create what we need to create and what we've lacked for so long in the nephrology community, which is really a research culture. We often look with jealousy to the oncology space, where they've developed precision medicines, and as we heard from Dr. Starr, that's where we need to go in the renal space. But we've also, obviously been very envious of their ability to develop a number of therapies and get them out to patients and really improve the lives of their patients. And they've been so successful in part, we think, because of their ability to create this research culture. And at least to date, we haven't been successful in doing that. But I think these groups are going to make us successful in doing that. I think the other thing I want to highlight in terms of how we got here is really because of the generosity and the willingness of at least some members of our nephrology community to really come together and share the data, right? It's great, it's nice, it's wonderful to do all this discovery work, it's wonderful to do all this work to try and understand biomarkers, et cetera, but it's not going to get us anywhere unless we come together and share the data. And I think there are both some wonderful successes of this in this this space, and in one of these I just want to highlight relates to a recent meeting we had with the National Kidney Foundation, Food and Drug Administration, where I am, and also the European Medicines Agency, which is the group in Europe that regulates drugs there, to try and again use the available data to let us come up with endpoints that we could use in trials that are reliable indicators of a treatment's effect on these later outcomes, and really do trials that are smaller and faster. I think there have been a lot of successes in terms of coming together and sharing the data. There are also some places where we haven't been so successful, and I think one of those is really the biomarker space for acute kidney injury. That's in many ways been a painful process to watch, and that I think for 10 years, if not longer, I'm sure much longer, there's been a lot of excitement and discussion about biomarkers for acute kidney injury that are actually detecting the injury to the kidney. But for whatever reason, it's really taken us a very long time to really get them used in clinical trials, to understand them sufficiently, and to get them used, hopefully, to benefit patients and used in the real world. So we need to work harder on that one, but we will get there. And with that, I'd like to pivot and focus on one very important initiative at the FDA and the CDER side. You heard about it, a similar initiative on the CDRH side, and that is patient-focused drug development, and really the importance of the patient voice in drug development and initiatives to incorporate. If you think about certain things or think back to history and how people viewed the world, sometimes you look back and you could say, well, how is it that they ever, neglected to realize something that was obvious? Copernicus, taught us that the sun needs to be placed at the center of the universe. And looking back, you could say, well, how is it that we somehow miss the fact that patients should not be really at the center of drug development? I'm not sure how we ended up where we are, but somewhere along the way, we lost sight of that very important fact. But fortunately, we have woken up to this issue for a variety of reasons. Just like the community is waking up to this issue. And so at FDA, we have really a team put together to really advance the science as relates to patient-focused drug development. So what do I mean when I say patient-focused drug development? What I mean, or how we've defined it, is a systematic approach to help ensure that patients' experiences, perspectives, needs, and priorities are captured and meaningfully incorporated into drug development and evaluation. And again, this is the key. As experts in what it is like to live with their condition, patients are uniquely positioned to inform the understanding of the therapeutic context for drug development and evaluation, right? So it's obvious, but somehow we lost sight of this. So what are the overarching goals of this initiative on the drug side? One of the overarching goals is to facilitate and advance the use of systematic approaches to collecting and utilizing robust and meaningful patient and caregiver input, and to use that to inform drug development in our decision making. Another goal is to encourage the identification and use of approaches and really best practices to facilitate patient enrollment and minimize the burden of patient participation in clinical trials, understanding that you have already so many other things going in your lives as patients. Not only do you need to take care of your disease, but you also need to live your life, that is critically important. Other goals include enhancing the understanding and appropriate use of methods to capture information on patient preferences and the potential acceptability of trade-offs between treatment benefit and risk. And I think Carolyn also spoke to this issue as well. Really are needing to understand patients' tolerance for risk and some of the uncertainties as well. And finally, identifying the information that's most important to patients related to treatment benefits, risks, and burdens, and really how best to communicate that information to support their decision making. So how are we doing this? So even though we recognize that including the patient voice in drug development is critical, We also know that if you talk to one patient about their experience with the disease, you've talked to one patient. And so you really need to develop the science around getting meaningful insight into how diseases are affecting patients, patients' tolerance of risk and benefit, because again, if you've talked to one patient, you've talked to one patient. Or rather, as Carolyn pointed out, if you talk to one person with the disease, you've spoken to one person with the disease. So some of the ways that we're trying to get at this issue is by drafting and developing, very much with the input of the community, a process that was outlined similar to what Carolyn outlined, a number of guidance documents that really articulate how we should go about collecting this patient experience data and really how we should use this information. We've also, over the years, had a number of FDA-led patient-focused drug development meetings, and these were primarily held between 2000 and 2017, where we came in and we listened to the patients within specific disease areas about where there was need, what it was like for them to live with their disease. And more recently, we've been very involved in a number of externally led patient-focused drug development meetings as well. And just to highlight some of those in the recent space, we've had really some very, I think, very helpful, very powerful and important meetings across a number of rare kidney diseases, C3 glomerulopathy, Alport syndrome in 2018. And this coming August, we're going to have another meeting that focuses on IgA nephropathy, another uncommon kidney disease for which therapies are urgently needed. And so with that, I just want to close and close with just these thoughts and comments. I think as you all know, and as I have increasingly appreciated over time, it really takes a village to successfully develop safe and effective treatments. And important work is going on to address areas of science where we don't have a sufficient understanding, and Rob very much highlighted that important work. I do want to point out that there have been a number of important scientific advances in understanding at least a number of the kidney diseases that our community faces. Again, want to just emphasize that there's also just been tremendous interest on the part of industry in developing treatments for these diseases, and so now our charge really is to create a culture that allows for these studies to get enrolled and completed. But finally, the most important point I want to make is, as the person who is the expert in the disease and who will ultimately use these therapies, patients need to be at the center of this. And so with that, I will end. Thank you.
Hello, everyone. Thanks for having me. I am not a scientist, so I'm a lobbyist. I'm a different kind of ist. So I'm here today to talk about Congress and the congressional procedure and how that fits in with what it is we've been talking about. So you've also already thought about the folks you've been listening to for the last hour or so. Those are federal government bureaucrats. Ew, really? Remember all those terrible stories you hear about the bad people in government? Tell me how bad these people are. Really? Wow, there are some great things going on, not only at FDA and NIH, but believe it or not, in Congress, relative to kidney disease, ESRD, and transplant. By way of disclosure, I think I can figure this out. Maybe. Ed? Where am I pointing? Oh, there we go. This is who I am. So I've already told you I'm a lobbyist. My job is to be a translator, to talk about to members of Congress and frankly to their 22-year-old staffers about what it is transplantation, right? Transplantation and what we need as a community. I represent ASTS, as Ed mentioned earlier. I am also, I should say before I dive into my topics, an unapologetic supporter of AAKP. Paul and Richard and Ed, huge fan, even if they didn't give me an award last year, which I'm very proud of. I'd still be a great fan of you guys. What I know from ASTS is that for many years of representing them, they have said to me, where is the patient voice? When we, the transplant surgeons, the transplant surgeons go up to Capitol Hill, why aren't we bringing patients with us? And this was one of our wonderful members who is no longer with us, and I adored, who said that to us over and over again. And so I picked up the phone and I called Paul and Richard and I said, when transplant surgeons go to Capitol Hill, they shall never be without patients. Can you help me? And they absolutely. So thank you for that. It is wonderful. It's the right thing to do. It's who the transplant surgeons are serving anyway. So the topic issues I want to talk about today is that, am I at the right place? Okay. Oh, I skipped a slide. Somebody stole something off my printer. All right, I got to go yell at those lawyers back home. We have very active and engaged sponsors in Congress for a variety of pieces of legislation and different kind of legislative activities in a bipartisan fashion, in a bicameral fashion. You don't hear that every day, right? Here is something you should be jealously thrilled about compared to lots of other things going on Capitol Hill, is that we in our space, in transplant, ESRD, and the kidney space, have bipartisan friends. Have any of you physicians or any of you clinical people met a Democrat or a Republican kidney? I don't think so. So the good news is we are working with those individual champions in various ways to get them to do things that are helpful to us. So if you know anything about Jerry Nadler, left wing as they come, right? New York, Democrat. Jamie Herrera Butler, Republican. From Washington State, literally the other side of the country. Senator Gillibrand, running for president, also from New York State. Cotton, Arkansas, right-wing Republican. What? Look at this mix of people. How cool is that? That's what you all bring to the table, and that's what we're able to bring to our bills, and there are many others. Dr. Burgess from Texas has been fabulous and really, really strong on a lot of these issues. So diving into, let me see if I can go backwards. There are three pieces, or, well, issue areas, let's call them. One is Living Donor Protection Act. I know you know this. Who knows this, right? Everybody knows about this bill. It's been around for a long time. And I'll explain that and I'll go into it. Here's the easy way to think of these three things I'm putting on the table. Living Donor Protection Act, love it. Need it, let's push for it. There's a bill in the House, there's a bill in the Senate. This is great stuff. Dialysis Patients Demonstration Act, or as Paul renamed it, DIPTA. Not so much, and I'll explain why the not so much on that one. And then immunosuppressant legislation, also been floating around for a lot of years. This one I'd say needs a little tweaking before we get to the place where we're ready to move it. So Living Donor Protection Act, love it, let's talk about it. Dialysis patients demo, not so great. Immunosuppressant drug legislation, we're working on it. So getting deeper, right, and I'm terrible at this. Thank you. I think for the Living Donor Protection Act, this is truly the best atmosphere in a decade to advance this piece of legislation. I talked a little bit about the sponsors and what wonderful, strange bedfellows they are, from the right and from the left. What happened a year ago is literally game-changing on the Living Donor Protection Act. And what happened, and I'll tell you the little backstory of this, is we were trying to figure out why the Living Donor Protection Act was not moving. And there are a couple things that served as serious hurdles in Congress to getting legislation moving that I will talk about in a moment. My firm came up with, and it wasn't me, someone else at the firm dug deep into the congressional record and found out during passage of the Family Medical Leave Act, there was actually a conversation, we call it a colloquy, between members of Congress saying, hey, I just heard about this thing called living donation. Is this something the Family Medical Leave Act would let you take off work for if you're a living donor? And the answer on the floor of the House of Representatives, back when the Family Medical Leave Act passed, was yes. So many, many years later, we yank that out of the congressional record, run it over to the Department of Labor, just in the last year, and Secretary Acosta and his staff wrote an opinion. And the opinion said, we're right. So you can now use Family Medical Leave Act without a question if you have a living donor to work for you. That's a very big deal. And that became game-changing in the congressional side, and I'll explain why in a moment. There's also been recently vocal and active commitment from this administration on transplantation and living donation. It isn't that anybody ever opposed us. It just happens to be this administration for many reasons, some of which I understand are personal, for Secretary Azar in particular. Transplantation, living donation is something they care deeply about, and they are taking particular steps to try to make movement in these areas, which I think is very promising. We often, as I mentioned, Richard and Paul at AAKP, also work very closely with two other groups in this space. I shouldn't tell you this, but we call ourselves the Super Friends. Remember, I'm dating myself. Remember that 70s cartoon? So we're the Super Friends. We've known each other for a long time. The other two folks involved in that are NKF, National Kidney Foundation, and AST. So Living Donor Protection Act, here is a summary of it. Basically what it does is it does two distinct things. It prohibits insurance companies, life, disability, and supplemental companies from discriminating against a living donor if they donate. So how does this play out? You need a donor, you find a family member or somebody in your church, let's say, and they're worried. What's gonna happen? What if I can't get insurance? And I might need insurance, I'm gonna be one less kidney. I have to fill out the form. The form says, have I had a surgery in the last year? That makes people nervous. That's what we know. It's what's known as a financial disincentive to transplant. Why don't we take away that financial disincentive to transplant? The best way it was described to me recently is when we were on the Hill with Richard and Paul and with our transplant surgeons in the last month or so, is that a transplant surgeon who sat down with that 22-year-old staffer and said, I have a patient, 55-year-old woman, tried to get supplemental insurance, filled out the form, checked the box, and the insurance company came back and said, we're doubling your rates. And she went, what? Do you have any idea what it takes to be a living donor? Thank you. You're an astronaut kind of health level, right? So this woman fought. She took her medical records and she marched them in to that insurance company and said, I'm healthier. I am much healthier than the rest of the population, or they wouldn't let me be a living donor. This is the kind of sense you have to talking to people, when you're working with Capitol Hill. And insurance companies, frankly. So this patient went to the insurance company, made her case, and they came back, I love this part. They came back and said, not only are we not gonna double your rates, we're gonna cut them in half. If insurance, those kind of insurance companies could figure it out, they would want living donors like you, right? Because you're a way better bet than probably most of the population. So that's what we're trying to sell. That's what we're trying to make people understand on Capitol Hill. So again, first part of the bill is removing those disincentives to transplant in the supplemental life and disability space, so people don't worry if they do get sick from something else, not their missing kidney, that they're gonna be able to take care of their families because they're gonna be covered by insurance. The second part is what I was talking about earlier, is the clarification that living donors can use FMLA for living donation, right? And then the third part is, once that stuff's done, we want HHS to tell everybody. AAKP in recent years, since that opinion came out in the last year, has probably done the most magnificent job of telling the world. So a year and a half ago, if you had Googled, does Family Medical Leave Act cover living donation? You would have seen a bunch of ads for lawyers, sorry, my brethren, saying, we don't know, call me. We can figure it out, we can help you get it. That's what it was just a year and a half ago. And if you Google it today, you will see AAKP, you're gonna see ASTS, you're gonna see Department of Labor, and you're gonna see that opinion that was written by the Department of Labor that says, absolutely, you're covered. It sounds like a little thing, but it's not. It's removing a significant disincentive for people who are worried they'll lose their jobs if they give through living donation. So, Why do I have that in the bill if I have an opinion letter from the Department of Labor? Why do I need that in the bill? We want it codified. We want it in law. No unclarity in the future. We need that in law. So Living Donor Protection Act, for those of you who aren't being helpful right now to get supporters, more co-sponsors on the bill, please, I strongly encourage you to ask your members of Congress, senators, representatives, to sign on. And if they're already signed on, please thank them. We've got to move this legislation. I think we have a very good shot at moving it this year. So here's the legislative status. In the House, there are 53 co-sponsors already. It's not bad. That's a pretty good number. But if you think of it, there's 435 members of Congress on the House side, so we need more co-sponsors. S511 is the bill on the Senate side, and this is a good number. Senators a little more judicious, if you will, on when they sign on to bills. But we have a lot, so please help us get more. Why the Department of Labor opinion is such a game changer is I'm getting a little inside baseball on the politics, but I think it's important here, or on the political process. The legislative staffers' fear has always been, if you quote unquote open up the FMLA, meaning put it on the floor, put it in a committee for a vote, for a hearing, for amendments, then everybody and their brother is going to say, I want FMLA when I stub my toe. I want FMLA for any possible reason. I want to make sure that I can take leave off of work. So there was a fear among these staffers and members of Congress that quote unquote opening up FMLA would hurt the prospects for this bill because it would just die of its own weight. What the DOL opinion does is it says, of course, that you can use FMLA. So in Congress, if there's already an existing opinion from an agency, we now have an argument to say we're not opening it up to anything else. We're going to put an FMLA bill, Living Donor Protection, which has FMLA in it, in a committee, in a process, in a hearing, and you can turn to everybody else who wants to change FMLA and say, oh no, do you have an opinion? Do you have an opinion from the Department of Labor? Do you have a record in the colloquy, between Congress? Because you don't get to be in that bill. Game changing. It helps us terrifically in the political process. The cost of a new mandate, particularly conservative Republicans or even conservative Democrats will tell you, it's going to cost so much money for those poor employers to let people take off 4 weeks, 6 weeks, right? I'm laughing too. To take off this time to be a living donor. Number one, the population is pretty small. We wish it was more. But the population is pretty small of the people we're talking about. We're also talking about super healthy people who are very, very healthy going forward after they give their donation. With the Department of Labor opinion, it's already theoretically happening. So the argument we've been making to Congress in about the last year has been, it's no new mandate, it's already an opinion. That's very helpful to us who work in the political world. There are also, and Paul, this better not have been you, somebody shoved a couple bills together years back, and in Congress to get through legislation, the parliamentarian assigns bills to committees of jurisdiction. Committees of jurisdiction basically means you get a chunk of law, Social Security Act, you get Public Health Service Act, you get a chunk of law, and it varies widely. But our bill happened to fall into 5 committees of jurisdiction. Imagine going through 5 committees with 30, 40 people on them to go sort through the bill, move the bill. That is daunting. And that's part of the reason this bill didn't move a couple of years ago, is everybody looked at it and went, you gotta be kidding. With this DOL opinion, 3 of those committees of jurisdiction have already told Jerry Nadler, one of our champions on the House side, we'll happily waive jurisdiction. Now that you have a Department of Labor opinion, move it past us. Hopefully that sticks. He's a chairman. That means we can move this bill faster than we have before. So again, game-changing, important development on the FMLA work. Moving on, I started with happy. So let's go with not so good. So the Dialysis Patients Demonstration Act. Some of you are old enough like me, and I love Bewitched. Remember the show Bewitched? Elizabeth Montgomery, and then there was the Dick York and the Dick Sargent, and I'm embarrassed I remember that. But, you know, her husband was Darren. And what did the evil witch mother-in-law call him? Everything but Darren, right? Durwood or something like that. It's how she made fun of him. This bill was billed by the people who were pushing it, who we don't agree with, as the Patients Act. It is decidedly not the Patients Act, in AAKP's opinion, in ASTS's opinion, and lots of other people's opinion. So, I don't know, probably 8 months ago, Paul said, why don't we call this what it really is? Let's do acronyms. Let's call it DIPTA. DIPTA? That sounds a little bit like Durwood to me, right? So I've been having a lot of fun, thank you, Paul, running around not calling this bill the Patients Act, because that's not fair, but calling it DIPTA. The most recent draft of this bill was released in September, and it has not had any congressional activity or floor action to advance this piece of legislation. It is yet to be introduced, but the sponsors are trying to seek to reintroduce it. Last session, these are the sponsors. If any of you live in those states, Missouri, Oregon, Indiana, Florida in the House, sorry. Talk to your members of Congress, and please tell them, and I'll explain in a minute why this legislation is controversial. In fact, I think this legislation was noticeably controversial to two members of Congress from New Hampshire and Colorado, in Shaheen and Bennett, who recently told us they're not going to lead this bill. They don't want to reintroduce this bill because we, our groups, convinced them this bill is controversial and would be harmful to patients. So, again, as I mentioned to you, this was introduced as the Patients Act and referred to as DIPTA. It would establish a new demonstration program. So a demonstration program, government gets to do a demo, not unlike an NIH study, a little different, okay, a lot different, but the government looks at something under a demonstration, and the way they structured their bill, and this bill was advanced initially just by DaVita, and since then Fresenius has jumped on, and their patient ally groups. On the other side of the equation, transplant surgeons, AAKP, SEIU, insurance companies, and lots of people in the transplant space who are saying no to the bill. What it would do and why it's so destructive or harmful is that this demo would form organizations, and those organizations would be led by the large dialysis organizations. So DaVita would lead the demo, okay? Let's see how that works out. And it would take all Medicare patients and put them into a capitated system where there would be more payment that we think you'd get because you stick people in dialysis and don't move them to transplant. So more money to the people who provide dialysis in a demonstration with no input from the transplant surgeons, and it is not really voluntary. People cannot get in and out when they want to get in and out of this demonstration. Other than that, how was the play, Mrs. Lincoln? You know, I mean, not good stuff. So what we had argued is some of the things I just mentioned: make dialysis providers responsible for transplant. It would disincentivize transplant via strong financial incentives, and those financial incentives are tenfold. So I don't care how nice you are, people who work at those large dialysis facilities, a corporate entity making decisions, and the difference between a tenfold difference in money, probably not going to make decisions always in the best interest of the patients. The severe limits on beneficiary choice, I've been educated by AAKP on this, and by Patrick G. and Richard and others who have explained to me what it feels like to sit in that chair and what your first 75 days looks like, and how clinically your brain isn't processing the way it used to process. That's not talking down to you, it's just a clinical fact that happens to a lot of people. So a bill that comes along and says, you can decide to get out of this program in the first 75 days, doesn't make any sense. Not if they were really talking to the patients. So listening to the patients, the transplant surgeons have said, frankly, hell no, we won't go. This is not something that is supportive of patients. It also, for my government friends, contradicts and preempts an existing program. It may not be perfect, it's known as the ESCO program, but it is an existing demonstration about how to do better for patients in the space of transplant and dialysis in particular, but also in transplant. That's a public-private partnership, everybody works in the same thing. Why would we preempt something that's already happening? So those have been our arguments against DIPTA. Immunosuppressant drug coverage. Everybody knows about this, maybe? Let me go into it. So when the immunosuppressant drug coverage was passed, and this was, I think it was AAKP or the predecessor of AAKP, who brought to a congressional hearing the Ways and Means Committee hearing a person to be dialyzed in front of the committee members. You can never do that again. Way to go. And what that did is it impressed those committee members with, oh my God, Medicare should be paying to take care of these people. So little known fact is there is no other disease that gets the same kind of treatment that ESRD does under Medicare. We should be jealously guarding that. It's important that we get that, and it's for all the right reasons. And from great political movement like that, to show people what difficulty it is. So, great. That's helpful. What's the next thing? Drugs come along. And when the drugs came along, we're like, oh, well, yeah, I mean, they had to come along, right? Immunosuppressant coverage has to be part of transplant, or you will reject the organ. So the drugs came along and they got better and better. And Congress said, now I'm going to pay for somebody to have a transplant and we're going to pay for the drugs. How long do we have to pay for the drugs? And the clinical answer is the rest of their lives. And Congress's answer was, oh, we only have enough money for 36 months. You okay with that? The people who were in the room at that moment said, no, we're not okay, but if that's all you're giving us, we'll take it. So that's been the status quo in law for over more than a decade now. Yes, you get your transplant under Medicare. Yes, you get covered 36 months of your immunosuppressant drugs. And after that, I don't know. Government's not paying for it. So we've been fighting for many years to try to get immunosuppressant drug coverage extended for your life and to have Medicare cover that. It's been very difficult to get past that because you can imagine the cost. So this bill was first introduced in 2003, with Senator DeWine from my fine state of Ohio, and reintroduced in the 114th session with Dr. Burgess I mentioned earlier. And the most active time for legislation is when Dr. Burgess was becoming chair. I think he might have been ranking. You're either in the majority or not, and I think at the time you might have been in the minority. He's an OBGYN from Texas and cares deeply about all manner of science and is a really great guy to work with, big fan of transplant. And he worked with us, and his staff went almost daily to CMS, so Centers for Medicare and Medicaid Services, and the Congressional Budget Office. Congressional Budget Office is the group that decides how much a bill costs or how much a bill saves. The Congressional Budget Office kept pushing back and saying, well, this is going to cost money. You're going to give drugs to people for the rest of their lives. It used to be 36 months, and you're going to give it to the rest of their lives. Well, we can't afford that. That's going to be a lot of money. So it stalled, and it got very frustrating. In the last session of Congress, we didn't even introduce the bill. What we're doing now to try to move that bill to a place of understanding and lower costs is kind of threefold. One is we're reanalyzing the original bill to make sure that it says the right stuff, that it makes the most sense, that it cares for the people it needs to care for, that it provides the coverage. The second step is 10 years ago, the immunosuppressant drugs were not the same. The cost was not the same either, right? Thankfully, they're going down, is my understanding. So our score from the Congressional Budget Office should have precipitously dropped. So that changes the equation. And the third piece that changes the equation is, again, this administration. This administration, with the help of some of our allies and friends around the room, have released one study, and they're supposed to release two, that says, in their estimation, having a kidney transplant as opposed to dialysis for a period of time saves money. It should save the federal government money. So if you talk to any transplant surgeon, you talk to any researcher in this space, they're going to tell you, We all know transplant saves money and lives. God forbid we talk about that. Saves lives and money over time. Dialysis is more expensive. I think it's about after 18 months. So why can't the Congressional Budget Office just say, oh, well, there's the science. The cost is lower if you get a transplant. Why don't we do that? Because that's not how the Congressional Budget Office works. They don't score what we call prevention. So all of us lobbyists and policy people have just been I know, stupid, right? Banging our heads against the wall, trying to figure out a better way to manage this. And in swoops the administration with not just one study, but hopefully 2 coming out soon from the Office of the Actuary, making our argument. Will that convince the Congressional Budget Office or not? I don't know. But a number of our groups, have put together a request for a proposal to go out to analytical firms to make an even better case. So we're on fire, again, on immunosuppressant drug coverage. This can't just sit for another 10 years and people stop getting coverage at 36 months. It didn't make sense when they put it into law, and it still doesn't make sense, and we are working very hard to try to fix that. So these are the stalls I just mentioned. Sorry I didn't flip. You can read them. I'll breathe for a minute. You can look at them. La, 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 la. Okay, ready? Okay. The administrative Office of the Actuary, by the way, hopefully is going to come out with this, we're thinking a week or 2, something like that. And these are our new opportunities. So with that, I will say those are the 3 biggies. The yes, we really like it, the no, we don't like it, and we're working on it. There are other things that we lobbyists and congressional people who fight for you and transplant surgeons and nephrologists every day are working on. I will not steal Todd's thunder from ASN and talk about Kidney X, but just to say ASTS is a huge supporter of that. There's also efforts at replacing lost wages that are happening through a system called the NILDAC that takes care of transportation costs and others for living donation. There is also great what we call appropriations language that has been advanced through Congress with a community of transplant and kidney folks who are also working on your behalf. There's a lot going on for you that I'm frankly lucky to be part of your team. So thank you.
First of all, thanks to our great speaker. One of the key messages of our Global Kidney Summit has been that the patient engagement should be at every level of healthcare system, starting at the stage of research and development, such as Kidney Precision Medicine Project, patient-focused drug development to legislative process. I'm going to start with a question for Dr. Starr, and then we're going to have our audience to have questions and comments. So the nephrology community is very excited about the Kidney Precision Medicine project and to have a major atlas as a reference. Is there a consideration to include the molecular signature of the kidney-specific cells isolated from urine? So to have a non-invasive test to act like a window to the complex pathology of the kidney diseases?
Thank you for the question. So to translate it a bit, instead of just basing this on kidney tissue, can we get the same kind of information out of the urine? That's actually one of our goals of Kidney Precision Medicine Project, is to be able to get to that point. Right now, because of funding considerations, we're not investigating urine cells. We're banking urine, so the specimens will be available. There are groups that are trying to do that. It turns out to actually be quite difficult to do what you just said. I just heard a presentation 2 weeks ago on exactly this. It's hard enough to dissociate the cells within the kidney, fragment them into chunks and into individual cells. In the urine, the cells are there because they've already left the kidney. They're being in the process of dying, so that you're looking at a population of cells that are almost dead to begin with. Not completely, but almost. So it's actually difficult. But the simple answer is yes, we want to get there. It's going to be harder than people think.
Thank you.
Hi, Rainey Reganathan from GW. One comment and a question for Dr. Starr. So you had a slide with a real lack of therapies for acute kidney injury and chronic kidney disease. For someone who treats glomerular disease, I'm lucky that I actually have more than just a few medications, but I will say that even in that field, there's a lot of stagnation and a lot that could be done in terms of drug development for diseases like lupus in the kidney, FSGS, minimal change, IgA, things that some of our speakers have already mentioned. On a practical level for the Precision Medicine Project, are the results of the biopsies available to the patients and the clinicians at the time or after the biopsy is done, or are they just banked for research purposes?
Thank you. So there are two parts to that. Let me answer the first and the second, or actually the second and the first. Return of results has been a major desire of the patients, participants in the study for obvious kinds of reasons. I've actually been working closely with CMS, Center for Medicaid Services, and the CLIA people, the people who kind of decide whether we can or cannot return results, to negotiate exactly what we can return and what we can't return. And those discussions are ongoing and actually moving forward reasonably well. There is a law. We have to be careful about the CLIA law. So we are going to return the biopsies, the biopsy reports, and the creatinines, essentially the CLIA, the things that were collected under CLIA. We're in negotiations with CMS right now, and I think in a couple months we'll probably be there, being able to return the omics studies. I think what the patients would like to see, as I understand it, is essentially to put on their refrigerator the equivalent of the ultrasound of their kidney, or the ultrasound of their, if they're pregnant, what the uterus looks like. So that's what we would like to be able to get to, and we're moving quickly in that direction because we understand exactly what participants want. In terms of glomerular disease, you didn't ask it, but I will answer it. We haven't included glomerular disease in Kidney Precision Medicine Project because we have two projects that are already doing this, as you probably know, Neptune and CureGN. So, I mean, at some point these things will converge, but at the moment they're separated mainly because of the patient populations are so different.
Thank you.
And this, I think the question was not asked to me, and it was a critical question. I just do want to highlight that at least for two of the disease areas you've mentioned, FSGS and IgA nephropathy, there's actually an explosion of clinical trials going on. And so hopefully you're enrolling your patients in these trials and you're aware of them. If you're not, I would look to the site, reach out to the NephCure Foundation, because they're really, the IgA nephropathy foundation is now partnered with them as well. And they're really, there's a dramatic push to get these trials enrolled so that industry doesn't leave this space.
Hi, my name is Shay Dadgar from GW. Thanks for the great presentations. I actually have two questions, one for Dr. Starr and one for Dr. Thompson. As you know, when the cells gets to the state of being necrotic, they often dead, and it's too late to target them for therapy. I was wondering if your technology allows to detect the cells at the stage of pro-inflammatory / pro-fibrotic state, so that we can use this information and directly target them for the therapy and translate it to clinical use.
Great question. The answer is we hope so, and let me explain. In an individual patient, what we'll probably end up seeing is a variety of cells, some healthy, some diseased, some undergoing repair, et cetera. There are new techniques called pseudotime analysis that allow you, at least mathematically, to look at what happens along that pathway within an individual patient. Okay. On a second level, we'll be able to do, once we accumulate enough patients, is to see where a patient is on average versus their peers along that kind of pathway. So the techniques are rapidly evolving, and you're right, dead cells are dead, but we hope to get information from the other cells in the biopsy. The other thing we hope to do over time, what I would love to be able to do, is a imaging-guided biopsy, to be able to take an image of the kidney, particularly with some new fibrosis markers that are being developed in a different part of the studies that we fund, that may work as in an MRI setting. These are cleavable compounds that will light up where the kidney is fibrotic on an MRI machine, and then go in with a robot and pick out, okay, we're going to take one good part of your kidney and one bad part of your kidney. So we'll know where the biopsies came from in comparison to the bands of fibrosis that are seen by MRI. So that's a little bit more space age, but probably not too far down the line. If you want to see an example of data not published from KPMP, but somebody who's working with KPMP, Andy McMahon Lab, looked at 50 to 100 or so kidney biopsies and did this pseudotime kind of analysis and showed that they could bucket patients into ones that would do well and ones that would do poorly a year later. So it's one of the best studies that I've seen recently, and this was done in ground-up tissue. This was not done on single cells. So I think the technologies are rapidly evolving, and we're aware completely of the kinds of questions that you're concerned about and trying to devise analytic ways around them.
Thank you. And the question for Dr. Thompson. So 20%, approximately 20% of the drug on the market becomes off the market due to adverse side effects. And some of the proportion of it, not exclusively, can be explained by pharmacogenetic, variability in the pharmacogenomic that patients carry. So the companion test becomes a routine for oncology treatment. I was wondering if you have any idea of where we stand as far as companion, pharmacogenomic testing as a companion test for the drugs that are applied to kidney disorders.
Right, so this is an area of a lot of interest, and really, hopefully, we're hoping also that the precision KPMP project that Rob has spoken about will ultimately also help us understand this issue better. I think in terms of understanding the genetics of some of these diseases and targeting populations, I think the one where we perhaps have the best example at this point is APOL1, which I think you heard about yesterday. If you were here yesterday, I was not, unfortunately, but that is really a very exciting discovery. We know, unfortunately, that the African American community has had a disproportionate burden in terms of kidney disease and its progression related to a number of factors, but it's now becoming increasingly apparent that some of this is genetic related. And I can say with much excitement that we now have some sponsors who are interested in going after that. So more to come.
Thank you, and our time is over. Thank you.
Thank you to our panel. Appreciate it.